This is experiment 14.3. We are looking at the different fixed slides on page 450. It says to look at the leaf cross section first. This is the leaf cross section under 100 power. As the book says, that seems to be the best place to look at it. I want you to notice it does look a lot like the picture in your book. Here we're to identify the upper and lower epidermal cells. Once again, the epidermis is the single cells across the top and the single cells across the bottom. How do I know that this is the top and this is the bottom? Were you able to figure it out? Because the Palisade Mesophyll is here and the Spongy Mesophyll is down here. So this means this is the bottom of the leaf and this is the top of the leaf. So this would be the upper epidermis, and this would be the lower epidermis. We are to identify the Palisade Mesophyll, which once again is your Palisade Mesophyll because you see it's like little pieces of a fort stacked together. And then the vein actually is here. This would be our vein. Let me just adjust this slightly so we can get it a little clearer. That's the vein. The spongy, actually there's a few veins here, aren't there? The Spongy Mesophyll, obviously this is the Spongy Mesophyll where you see the holes or pores in it. And then the guard cells and the stomata, let's adjust so we can maybe zero in and see if we can find. I'm going to go down here. Let's see if this is a stomata. I'm going to go on in here because that's an air hole, obviously, because there's a lot of space here. So those might be the guard cells. I'm going to go ahead and focus in at 400. Let's see if we can get a better shot at this. There we go. And so at 400 power, it would be my guess that that is the stomata, and those would be the guard cells. I'm going to move along here at 400 if I can. None of them appear to be open, but that again I would say right at the pointer would be the stomata and the guard cells on either side of it. And so remember, when there's plenty of water around, they swell up and open, and when there's not, they shrivel and they close, so that the plant does not photosynthesize. So I would think that these would be found most readily right along there. Let's go back. And I'm just going to give you a good swing of this leaf. Cross section. Once again, right there at the end of the pointer is probably guard cells with the stomata. And here you've got full length of it. I would show it to you on 40 power, but for some reason on this camera that you're looking through, when I go to 40 power, we lose the good view of it because the light becomes overwhelming. And so I'm showing that to you so you can see the light becomes overwhelming in that case. So it's better that we look at it at 100 power and that I zoom you back and forth. So that's our leaf cross section, and we've identified all the parts that we were supposed to there. You're supposed to draw this and label it, and this one can be done here, or you could do it from the picture in your book. As I suggested in class, I wouldn't draw all these circles. I would draw a representative of the Palisade Mesophyll, maybe like draw this section through here and just do representative circles in the different areas, rather than doing all of these circles. And then using colored pencils will definitely make your presentation and your observations much nicer. Let's go on then and look at the cross section of the ranunculus root. This is the ranunculus root cross section at 40 power so that you can get the whole root in there. We are supposed to find the epidermis, and that would be on the outermost edge. Then we have the cortex, which is the in-between. Here we have the endodermis. You can see the central region. And then in the central region, we are supposed to find the xylem. So I'm going to focus in at 100 power now on this central region. This is focused in at 100 power on the endodermis, the xylem, phloem, pericycle, and so I think the larger holes are our xylem, and the smaller groupings are the phloem, and the pericycle, I'm using the figure 1411 on page 445 to try to help me. The pericycle, if this is our X, would be in this vicinity in the crosses of the X. And so that would be the vascular portion, the vascular tissue in our Dicot root cross-section. This is our Z-maze root cross-section. And so this is our example of a monocot root cross-section. Once again, the epidermis is on the outermost edges. The cortex is between the outer edge and the vascular system inside the root. And so that central region that you're seeing there, it's a center on the vascular tissue, which is just on the inside of the endodermis, and increase our magnification to see if you can distinguish the xylem from the phloem. And a monocot root has a section of pith. So let's go ahead and we're going to try that. We're going to go up to 100 power. All right, so here we have the vascular system in the center of the Z-maze, which is our monocot. And you see the endodermis, which is around the outside of this. And then it mentions that there might be pith here, and I would guess that this portion in here would be the pith, because once again the pith will eventually almost seem hollow, and it's usually at the center. And from what I've read, it's for initial storage of fluids. This would be the xylem, the bigger holes, and the phloem, from what I've read, is the smaller holes usually. And so that would be our xylem and our phloem in our Z-maze cross-section of the root. Now we're to the Z-maze stem cross-section. And I want you to notice, as I'm going to pan around for you, this is at 40 power. And so there would be your dermal tissue, your epidermis, and the cortex is just the portion in between. And all of these are fibrovascular bundles. All these that look like little faces are fibrovascular bundles. And I can finally really here it's better to see how they look like little monkey faces than it was in class. You can almost kind of see a little monkey face. There's his eyes, there's his nose, there's his little mouth. And so that would show you the fibrovascular bundles in the monocot, which is what Z-maze is corn. And a matter of fact, let's focus in, and I want you to just notice there's no donut thing going on here, that these things are everywhere. These fibrovascular bundles are just distributed everywhere throughout the cortex of the stem. And then we're going to go ahead at 400 power, focus in on one of these little faces, which is the fibrovascular bundle. So actually the pointer's on the phloem. So these are the xylem, and this is the phloem in that smaller region. Oh, and this big one here is an air space. So from what I'm going by the picture on page 446, figure 1412, it tells us that this is an air space, this is the xylem, and this is the phloem. And so those are the fibrovascular bundles. So to do another one, this would be air space, xylem, phloem. And so that would be, I'm going to go back down to 100 power. That would be your monocot, herbaceous, stem cross section. I just want to pan over so you can see it. So we identified all of the things he asked for there, the ear space, xylem, phloem, fibrovascular bundle, cortex, and the epidermis. So now let's go on and observe the ranunculus stem. At 40 power, as we come into the Dicot stem cross section, I want you to notice there's our Dicot donut. See, you have it, the fibrovascular bundles around the edges. You have the big open pith space in the middle. We have the epidermis along the outside edge. You have the cortex in between, the large pith space in here, and then we have all these fibrovascular bundles. Once again, notice that these fibrovascular bundles don't have the monocot monkey face going on. So let's go ahead and go to one of these fibrovascular bundles, and we're going to go ahead and focus up to 100 power. So we'll bring that into focus. And what we see here, we're supposed to identify the different parts of the fibrovascular bundle. So as we look at this, the xylem are these bigger holes, the phloem is in this area, and this is the vascular cambium, which can still become whatever the vascular tissue needs. If it needs more xylem, it'll become xylem, so it's undifferentiated. If it needs more phloem, it'll become phloem. It can be whichever. So when we look at this one, we see that there's the xylem, there's the vascular cambium, and there's the phloem. And you are supposed to draw those. This is a close-up of your epidermis. This would be your cortex. Once again, don't get crazy and draw every circle. I'm going to go back here for a minute to 40 power just so you can get a big picture of this again. And so here you see the whole characteristic donut-looking shape with the fibrovascular arrangement being around the outside edges and the large open pith space in the middle. And so you are supposed to draw those and label them and identify them for this particular lab, and that would be very helpful for you to do that before you take your test.